Dr. Kelli Barr is a professor in the biology department, and she teaches a course called Virology. And Kelli is here today to share some information about viruses with us.
So, okay. Hi, I'm Kelli. It's nice to see you all here. I'm so excited to see so many younger people because it's your generation that's going to start learn the most about viruses and start turning things around. So I'm going to start sharing my screen and let's get going. So virology is a ginormous discipline, and I can't go over everything in the time I have, but what I want to do is just give you a basic overview about how viruses work and how they're spread, and then I'm going to take some of these concepts and then apply them to the COVID outbreak to just give you some perspective with what's been happening. So, who am I? I want to give you a little background about me, where I came from. I'm from Iowa. I went to Iowa State and I got my degrees in English and classical studies. But after that, I really wanted to help people. So I went and I got a PhD in plant, insect, and microbial sciences. And in there, that's where I learned about viruses and how microbes can affect plant, insect, and even human health and animal health. And so I want to give you some background. When I'm talking, a lot of the information I present in this presentation comes from the textbook I use when I teach my virology class at Baylor. And this picture just is of the authors who wrote this textbook. It's an amazing resource if you want to get your hands on it. So a little bit about my background. I've been studying viral outbreaks for over 10 years. I studied the mosquito and tick-borne viruses. And so I've been watching the dengue outbreaks in Key West and Pakistan. I work a lot in Pakistan. And I've worked on the chikungunya and West Nile outbreaks there. And I worked with healthcare and other researchers there to develop better diagnostics and testing for the people, because when viruses emerge in a new region, they oftentimes have different symptoms. Sometimes they can be more severe, sometimes it's less severe, but they usually have different symptoms or presentation that can make it difficult for doctors to identify. So I help with that. So let's talk about viruses. So they are everywhere. I mean, everywhere. They infect every living thing. Every time you take a breath, you eat, you are consuming thousands of virus particles. They've co-evolved with us over millions and millions and billions of years. They're part of our DNA. And at any given time, I could take a blood sample from you and find hundreds of different viruses in your bloodstream. So not all viruses are bad, but we do know that they're very, very, very small. On the head of a pin, it's about 2 millimeters across, you can fit 500 million rhinoviruses. Rhinoviruses cause the common cold when you get a head cold and it's stuffy and you're sneezing. Those are rhinoviruses a lot of the time. So when you're sick with the virus and you sneeze, you shoot out enough virus particles to infect thousands of people. So it's important to note, I mean, a lot of us get freaked out when we hear about virus infection, but the truth is that most viruses we are infected with, they have no impact on our health. And so they're not all bad. Most of the virus we're infected with is just passing through. So every time we eat a fruit or a vegetable, you're eating tons and tons of plant and insect viruses. There's one study that found that in one serving of coleslaw, which is cabbage, it contained 1 billion virus particles that were pathogenic for an insect, which is this cabbage looper right here on the right. The virus doesn't hurt us at all, but it will kill an insect. So that's pretty cool. So it's important to note there's a lot of talk about viruses in the media. Viruses are not alive. It's just basically a protein coat surrounding nucleic acid. So viruses can be either made up of DNA or RNA. So it's important to know they can't move, they can't think, they're not crafty, they don't evade, they can't even grow. They need to live inside a host cell to get the things they need to assemble new particles. And once a virus is outside of the host, it can't replicate, it can't make more copies, it can't grow. So if you sneeze on your shirt, virus can't grow and multiply on your shirt. It's just there and it usually dies. So why should you care about viruses? Well, if you combined all living things on this planet, viruses would still outnumber us by 10 to 1. They drive global cycles, and a lot of viruses can be beneficial. We've identified a lot of beneficial interactions between viruses that infect plants, animals, and even humans. But what I like most about viruses and what makes me so excited to research is that viruses evolve really fast and they really can evolve quickly into new pathogens. And what I like best about my research is I study a lot of viruses that can jump between species. So there's a lot of viruses that can infect birds or pigs or bats and can jump to infect humans or other animals. I also like the vector-borne, the mosquito and tick-borne diseases because they have to live inside an insect and inside a human. And so the virus has to be pretty genetically advanced to be able to survive in those two different environments. So let's talk about viral infections. There are two basic patterns of infections. You get acute infections. That's like when you get a cold or the stomach flu. It's you get sick and then you get over it and then you go on to live your life. Then we have latent and persistent infections, and that's where you get infected with the virus and you're infected for the rest of your life. And that's what we see with like herpes virus, HIV, hepatitis. An important part of the virus life cycle is the incubation period. So the incubation period is the time when you get infected and the virus is making more and more copies. And this is before the symptoms of disease are obvious. So you might be feeling a little off or a little tired, but you don't really realize you're sick yet. So your immune system is starting to respond. And at this time, some viruses can be transmitted even before you feel sick. So measles virus, before you develop any signs of disease or a fever, you've already been spreading it around for a couple days. So when a virus reaches a certain level, we get the infectious period, and that's where you can transmit the virus to other people. And a lot of times this is where we start to feel sick. So there's vocabulary that's important. A symptom is what you feel, like a headache, or nauseous, sick to your stomach, but signs of infection are what other people see, like a rash, coughing, sneezing. So let's talk about viruses in a population. So we talk about the burden of disease. And here I'm presenting a graph of influenza just because we know so much about influenza virus. Since the pandemic, the influenza pandemic in 1918, that motivated governments to institute continuous surveillance and testing so we could be prepared for a new pandemic. And so that's the data I'm presenting here. So for any virus infection in a population, you have people who are infected. Most of the people, for most viruses, it's different for every virus, but you have a portion of the people who are infected, but they don't feel sick. They either don't get sick or they don't feel bad enough to go to the doctor. Then of the population that's infected, you have a portion of people that feel sick enough to go to the doctor. So after that, you're at the doctor examines you. There's an even smaller portion of those individuals who the doctor feels that they should go to the hospital. And of those people that are hospitalized, there's even a smaller portion of those who die. And these proportions differ for every virus, but for flu, this is what it's like. And it looks like COVID's about the same. I know we're seeing a lot of numbers about this pandemic, but the only numbers we are seeing are the people who are hospitalized and the people who are dying. So we don't know how many people are out there sick with COVID or infected with COVID that are just don't feel sick enough to either go to the doctor or they have no symptoms. So the percentage of infections in our population could be much higher than what we're seeing. So how do we measure virus infection? There's a lot of tests we can run. We can look at how the virus damages blood cells. We can look at them under microscopes. But the most testing we do in the doctor's office in our labs, we use RT-PCR to look for viral nucleic acids. So we look for viral DNA or RNA. And then we can also test your antibodies to see if you've developed antibodies specific for a virus. So up here you see on the top right, this is an ELISA plate. So you've probably heard vocabulary in the news about this COVID outbreak. They're doing ELISAs, they're looking for IgG or IgM antibodies. IgM antibodies are what your body produces very early in the infection. And then the IgG antibodies come later, and they are more specific to a certain pathogen. And so up here on this ELISA plate, you run your samples, and if you're positive for having antibodies to a virus, you'll change color. It's usually blue, and that tells a scientist that you've been infected with the virus. Another thing we like to use are lateral flow assays. They're the next one down, and these are my favorite things to use because they are fast. So if we've all been sick with the flu, and probably all have gone to the doctor, and or even sick with strep, and they swab the back of your throat or they swab your nose, then they what they do is they mix it with a little fluid and they put it on these lateral flow assays. And if you have virus antibodies in your blood, that'll react and turn a different color. It makes it like a line, like on a pregnancy test. I like these because they're fast, they're cheap, and you don't need a lot of training to do them. So these are the tests when you hear that they're approving pharmacists or other types of health professionals to run testing for COVID or flu, it means they're going to be using a lateral flow assay like this. These are great because they're easy to pack along, and they're affordable for a lot of places where they don't have the infrastructure to do PCR or ELISA testing. So I'm going to stop right here and for questions before we move on.
If anyone has any questions so far, go ahead and type them in the chat and we can read them to Dr. Barr. Sarah asks, what does RT-PCR mean?
Okay, RT-PCR stands for reverse transcriptase PCR. And so there's an enzyme called reverse transcriptase, and what that does is it turns RNA into DNA. So what we do is and PCR stands for polymerase chain reaction. So if you look at the diagram on the left, what we do is we take a blood sample and we isolate all the RNA out of it. And then we add some chemicals to it, we put it in a machine, and we add the reverse transcriptase to turn that RNA into cDNA. Then we run the PCR, and what that does is you heat up a sample and that breaks apart the DNA, then you cool it down, and the DNA will come back together. But when that happens, there's enzymes in there called polymerase that synthesize new strands of DNA that are copies of a template strand, which is the parent strand. And so we can take one strand of DNA and amplify it by heating it up and cooling it down, and heating it up and cooling it down. So we can take one copy and make millions of copies to give us a bigger signal to see whether or not you're positive. Did that answer your question?
We can move on to the next one. A couple of people have asked, what is the best way to tell if you have COVID-19?
Well, there's different tests you run depending on the stage of infection. So if you are sick and feeling like and running a fever, RT-PCR is going to be your best bet. But if you were sick and you think, I bet I had COVID-19, you would do the antibody test like the ELISA or the lateral flow.
Another person asked, have you ever studied a virus to which humans did not develop antibodies?
No.
And then we'll do one more question from Alan. He asks, are there tests being used that aren't as reliable as we hope?
For COVID? Do you think this is relating to COVID?
Yes.
Yeah, okay. So when a virus emerges, it takes several months for us to develop these assays to identify antibodies that are specific and can be used with a broad population of people. So we're all different and we all react to viruses the same way. So we have to make materials that will, that a bunch of people will react with. Also, what's tough is a virus like COVID is RNA. So it has a really, really, really high mutation rate. So we have to generate materials that will react with COVID even if it's mutated. And so that takes months to do. And then we have to find infected people to test it with to make sure it works. So for any virus that emerges, there's like a good 6-month lead time before you get really good diagnostics. So we have the methods in place for building good diagnostics, but it just takes us time and a lot of samples from people and a lot of viruses so we can make the best test we can. So right now, I guess the answer to your question, the tests, they're working, but they're going to get better as time goes by.
Well, so if we didn't get to anyone's questions now, we can hang out after the chat and we can answer them later, but we'll go ahead and move on with the presentation.
Okay, let's go. So I'm going to address a lot of common questions about virology that I get asked. How do I get infected with the respiratory virus? So the most common way is through just breathing in virus particles. You can also get infected with them by touching something that's got a virus on it, like doorknobs, cell phones, things like that, and then you like itch your eye. So what happens is the virus gets, I'm going to talk about the way it gets in when you breathe. So you breathe in the virus and it sticks to the mucus in your sinuses or the back of your throat, and it starts replicating in your cells there. Now, different respiratory viruses live in different parts of your respiratory tract. And this graph shows some of the different viruses that lived in the upper respiratory tract. Those are the viruses that give you sore throats, you lose your voice, or you get stuffy, runny nose. Then there's viruses that like to live in the lower respiratory tract. That's down in your lungs when you get those chest colds. Flu virus does that. Measles virus does that. And so do these pathogenic coronaviruses like SARS, MERS, and COVID. So how do respiratory viruses spread? So it might surprise you to learn that the most infectious particles are spread just by breathing and speaking. And if you think about it, it makes sense. When you're sick, you spend a lot more time, you're always breathing and you talk to people, but you may not sneeze or cough a whole lot. So the way this was discovered was, if you look at the picture on the right, this lab took a bunch of people who were sick with the flu and they put their faces in this machine. And the scientists had them, recorded them when they sneezed or when they coughed or when they were talking or just sitting there breathing. And that apparatus measured how much virus was being produced by the individual. And this is where they found that, one, most virus comes out when you're speaking, and then two, that wearing masks can actually prevent people from spreading disease, and it can offer some sort of protection. So these are just surgical masks. So if you look at the picture on the bottom, so when you cough or you sneeze, the droplets you produce go out, about 5 feet from your body. And so this is where social distancing comes in. If you're like 6 feet away, you're really not going to get in contact with any infectious droplets from people coughing or sneezing. But when people are talking, you get these micro droplets that circulate in the air, and depending on how humid it is or just the environmental conditions, those viruses can float around there for hours. So that's where masks come into play in helping prevent the spread of disease. So why do we have outbreaks and pandemics? There's several reasons. There's several drivers for infection, and you might be surprised to know that climate change is a major driver for infection. The ocean temps have gotten a lot warmer, and that's changed how we have our global climate cycles of El Nino and La Nina. that has led to desertification. So places that used to have a lot of rainfall, like in California, are turning into deserts. And then areas that didn't see a lot of rainfall in the past are starting to flood. So when this happens, you have major disruption of ecosystems, and animals, insects, and people are moving around and living differently than they used to in the past. Another infection driver is land use. Population is exploding, and we're living closer and closer together in urban and highly populated areas where you have a lot of people living closely together. Really increases the risk of disease transmission of any sort. Another big infection driver that people don't think about is birds. So birds are responsible for the majority, if not all, of our influenza pandemic. So migratory birds, they don't respect borders. They go wherever they want. They also bring with them insects. So ticks will get on birds, or mites or lice get on birds and bite them, and then the bird flies away and it can take those bugs to new regions. The biggest driver of infection and the cause of pandemics and epidemics is just human movement. If you look at these figures, I have a picture of shipping and transport, so boats just moving across the globe. Red and yellow indicates more ships travel that way. Next down, you have all the flights that you see, and you people, we can just travel around the globe in the blink of an eye. But another important driver of infection is the movement of refugees and people as they move slowly across the land. So if we know what's causing pandemics, why can't we stop the spread? And the biggest factor is lack of funds. It costs a lot of money to develop assays and tests and pay researchers to find things out. A big one is denial. People thinking it's not going to happen to me, I never get sick. You have non-compliance, anti-vaxxers, people who don't follow the rules, like shelter-in-place orders when they come out. And then diagnostics and testing can be hard. We may not develop them in time. And we don't have diagnostics for every virus. There's thousands of viruses out there that can infect humans, and we don't know which one's going to cause the next pandemic or epidemic. So when an epidemic starts, we always have to scramble to develop new diagnostics and tests. So if we can't stop a pandemic, why aren't there more vaccines and antivirals? This is a problem. So let's first go over how vaccines work. You go to the doctor, you get your injection of vaccine, and then your body mounts its initial immune response. And that's where your body makes a whole bunch of antibodies that are specific for the virus you were injected with. And then those antibodies go away. They still remain there in the background. They're like surveillance. They're going around looking for that virus in your blood again. So sometime down the road, if you do get infected with that virus, your memory cells are there, and right away they start producing tons of antibodies to clear that infection so you don't get sick. Vaccines work because of a concept called herd immunity, and that's where we have a whole bunch of people vaccinated for the same thing. So we can stop virus transmission. If you look at this figure at the bottom, the red dot is a person who's sick with the virus, and the black dots are people who don't have a vaccine. And so you can see an infected person can spread virus to a whole bunch of people. But as soon as you start vaccinating people in the population, those are the green guys, you can see they can block the infected person from transmitting to people who aren't vaccinated. And then when you have a lot of people vaccinated, virus can't spread at all. So the number of people in a population that need to be vaccinated are different for every virus. I'm sure you've heard in the news this term called R0 they keep talking about. The R0 represents the number of people an infected person could infect. That's a tongue twister. So let's go through some viruses and I'll explain it. So smallpox virus, it has an R0 of 3.5 to 6. So what that means is if I was infected with smallpox, I could potentially infect 6 other people with smallpox virus. And if I want to stop that transmission, 80 to 85% of the population needs to have a smallpox vaccine. or it'll turn into an epidemic. Measles virus is one of the most contagious viruses we know about, and it has an R naught of 18, meaning one infected person can infect up to 18 people. And that's why they want us to have 95% of the population vaccinated. Some other viruses you might have heard about that we get vaccinated for is chickenpox. That's a respiratory virus, and that has an R naught of 12. The regular flu we get every year, that has an R naught of 2. And we think of flu as being very contagious. Now, COVID-19, since it's new and we don't have the data of inapparent and subclinical infections, the R naught value is still kind of not solid, but we're thinking it's somewhere between 2 and 5. So it's at least as contagious as flu, but we're thinking it's probably much more contagious than flu. Okay, so requirements of an effective vaccine. So if you're going to give someone a vaccine, it has to be safe. You can't make the person sick, have any adverse effects, or no one's going to take it. The vaccine has to give you the antibodies you need to protect you from future infections, and that protection needs to be long lasting. People don't like going in every year and getting shots, and some people don't have access to healthcare where they can go on a regular basis and get vaccines. The most important driver of an effective vaccine is its cost. The WHO wants us to produce vaccines that cost less than $1 per shot, and that's so people in developing nations can have access. So the other things we need to consider when we're making a vaccine, because we have to keep producing more and more and more, is that it needs to be genetically stable. It has to be the same from year 1 to year 2. And then it also needs to be able to be stored for a long time so it can make it out to people who live in remote areas. So, okay, understand why we have so few vaccines. Then why are there so few antiviral drugs? And it might surprise you to know that there are less than 100 antivirals available on the market. We have hundreds and hundreds of antibiotics, but the antivirals we have are mostly directed towards treating persistent and latent infections like HIV, herpes, and hepatitis. So when we're making an antiviral compound, it's really difficult because it needs to block infection completely. Viruses have a very high mutation rate, and if you don't block infection completely, resistant virus is going to grow out of there, because you're going to have resistant mutations, and if you're not blocking at all, they'll grow up. Another problem is that because viruses rely on you to replicate and reproduce, a lot of the antiviral compounds are also adversely affect you, and many of them have very horrible side effects that we just can't allow people to experience. Another problem we have is that not all viruses you can't grow them in a lab. And then a lot of these viruses, they don't have an animal model. So for human disease, if we're going to make a drug, before the FDA will consider approval or phase 1 and 2 trials, you need to prove that the antiviral works in at least 2 different animal models. So you have to say it works in mice and it works in rabbits, or it works in mice and it works in monkeys. It doesn't matter. It has to be shown in 2 different animals. And most viruses only replicate in 1, maybe 2 hosts. The other problem is most of the viruses we want to develop antivirals for acute infections, they're too dangerous to work with in the lab. So like Ebola and Lassa virus, those are BSL-4. There's only a few labs in the world that are qualified to work with those. And so these are problems. Other problems are from when we discover a potential compound that has antiviral activity, there's a lot of steps that take a lot of time. We have to prove that it's not toxic in cells. Then we have to prove it's not toxic in animals. And then by the time we get to the human studies, safety studies in humans, most of those compounds we realize have horrible side effects. Nausea is a really big one. And a lot of times they can have neurological effects, and we don't want that. And so you might start out with 100,000 compounds, but by the time you get to FDA approval, you might have 1. And all of that research and all of those studies can take 15 years. And $100 to $200 million to produce. So it's really, really, really hard. So a problem is that acute infections, they're usually a short duration. You get infected, you're sick for a week, maybe, and then you're feeling better. And so, but by the time, if you remember back to the incubation period, by the time you start feeling sick, it's too late for those antivirals to reduce your viral load and do anything to reduce the symptoms of disease. So antivirals, you need to give them the minute you start feeling sick. So next year at flu season, when you start having a fever or feeling like you might have a flu, you need to go to the doctor that day and get your antivirals. Because there's so many acute infections, it would be nice if we had a broad spectrum antiviral like we have broad spectrum antibiotics, but we just don't have any. And then we don't have a lot of rapid diagnostic agents for use when you go to the doctor. So we can do strep, flu, mono. There's just a set of maybe 5, but they're also bacterial diseases. So you need to have the right antiviral paired with the correct virus to get antiviral activity. You just can't take Tamiflu if you're infected with rhinovirus. That's not going to help you. So resistance to antivirals, this is a huge, huge, huge problem. So if you make an antiviral, resistance is going to develop because viruses have such a high mutation rate, all viruses. And so this has been a real problem with treating people with HIV and hepatitis with antivirals, because you need to take those medicines every single day. If you miss a dose, virus will replicate and will produce resistant mutations just through replication error. And that's how resistance develops. And so if you become resistant to an antiviral, we don't have a whole lot of alternatives to give you. And so you end up just getting sick and getting the disease. So these graphs show you the development of antiviral resistance. The top one is for flu, and you'll see it's just for one flu season. And so last year, by the end of flu season, over 85% of flu infections were resistant to Tamiflu. That's from people going into the doctor too late and not taking all of their medicine. The middle graph is HIV, the development of resistance to a variety of anti-HIV drugs. And then the bottom graph shows how over time resistance to antivirals has developed for a bunch of different antivirals. So, okay, we don't have a lot of vaccines and we don't have hardly any antivirals. So how can you protect yourself from infection? So you can do a lot. You need to be proactive. You need to wash your hands. Don't touch your face. And this is so important. Don't eat from communal sources. If you see a candy dish anywhere, don't eat the candy. They've done studies where they go and they swab the candy that's in the dish, and we've isolated tons of different viruses from that. I mean, we all assume we're all washing our hands after we go to the bathroom, but you don't know what people have touched between hand washing. So don't eat out of the communal food sources. Eat a healthy diet, take a multivitamin, get exercise and plenty of sleep, because that'll help you have a healthy immune system. And having a healthy immune system is actually your best defense from getting sick. When respiratory viruses are going around, practice social distancing. Just keep your 6-foot perimeter and don't be afraid to wear a mask. If you're sick, please wear a mask and prevent spreading viruses to other people. But if you're worried about getting sick, you wear a mask. It's no big deal. So what if this really interested you, and I hope it did. We need more virologists. How can you learn more? Well, here's some sources. These are my favorites. There is so much fake news out there, and I'm a professional virologist, and even I have a hard time sorting through it. There's research being thrown out there left and right, and not all of it's good research. And so even me, I always go to CDC or the WHO. They have accurate, reliable information. You can also use your state or government Department of Health. They're going to have the best real information for you. And then if you're just excited about viruses and want to learn more, these are my favorites. There's Microbe TV. It's a channel. It's science TV channel, and it's programs produced by scientists like me. There's the podcast This Week in Virology, where the host talks to a bunch of different virologists about different topics in virology. And then there's Dr. Racchianello's YouTube channel. So he has a lot of information out there too. So, but that's all I have for today. I hope you learned a lot, and now I'm happy to take any questions.
Cool, so if anyone wants to hang out and listen to our Q&A session, we'll hang out until about 11 o'clock to answer questions. So let me start from up here. Sophia asked, does each virus strip test change different colors for different viruses?
No, it's usually all turned blue. We'll have a different test strip for a different virus or bacteria, but they all turn blue. They might turn purple.
Someone asked, can you verify the accuracy of the testing kits for COVID? I was told that there's a 1 in 3 chance of false negative.
There's several tests being used that have been developed by several different companies, so I don't know which one you're talking about. The tests that the CDC and the WHO are recommending that people use, they're very accurate.
Liz asked, when you sing or shout, do you spread more droplets or do they travel farther?
They travel farther because you're breathing heavily, but those would be categorized in those micro droplets.
And how does the virus transfer from bird to human?
Through respiratory. So this happens all the time with flu, where you have people working on duck farms or chicken farms or a poultry farm, or even it's happened in swine farms where you're all breathing the same air, and that's how it happens. So this COVID, we believe it was transmitted by bat. You could have gotten it from eating undercooked meat. You could also get it from going in caves. That's how people got infected with Marburg virus, is just through inhabiting the same area and somehow coming into contact, whether you touched a surface and touched your eyes or touched your mouth. That's a way to get infection. You don't always have to breathe it in, but if you get the virus on part of your mouth or up in your nose, you're inoculating your mucosal epithelial cells, and that's where the virus likes to replicate.
Cool. Sarah asked, how many viruses do we know exist?
So thousands. So with, you might have heard about next generation sequencing. And so what that does is you can take a sample of someone's blood or a sample of water. And what it does is it identifies all the different DNA sequences that are in there. And so when we've done this, we have identified thousands and thousands of viruses, but not all of them are pathogenic. We know of about 500-600 that are pathogenic for humans, but I think we've discovered several thousand viruses. And you can compare that, we only know of a couple hundred bacteria.
Charlie asked, he said, I heard that there were 2 different branches of COVID-19, X and Y. Is this fake news? If not, do we need different vaccines for the X and Y? Okay, so yes, there's 2 different lineages of COVID virus. You have alpha coronaviruses and beta coronaviruses. So the alpha coronaviruses, they're associated with just like your regular old head cold. They have different, a little bit different virology or biology about them. I'm not going to get into it. But then you have the beta coronaviruses, and that's where we see the MERS coronavirus, SARS coronavirus, and COVID. So yes, if you were going to do a vaccine, it would have to be specific for the alpha or the beta coronavirus. You can't get a vaccine that'll cover you for all coronaviruses. They're 2 different genetically. Okay, next question. Are there standard specifications for reusable masks?
This is a tough one.
there are several different types of masks made by several different manufacturers that have different levels of infection protection. And if we're talking about for personal use, if it's a cloth one, I would just wash it every day. If you have reusable ones like the ones we've all picked up at Home Depot that protect you from the dust, my husband works at a hospital and they have to reuse their mask for the whole week. And so what I have him do is get a wipe to either spray it with some Lysol or 10% bleach or some sort of disinfectant. You can wipe it down with a Clorox wipe, or you can spray it when you get home.
This question says, is it true that COVID-19 has mutated in 3 sites of DNA?
Okay, so that's COVID, all viruses mutate. Every time a virus replicates, it mutates. And sometimes those mutations can be bad, and sometimes those mutations don't mean anything. But all coronaviruses are mutating all the time. And that's when you have people saying the COVID virus mutated, of course it did. All viruses mutate. And so it's nothing to be concerned about. People have been studying coronavirus for years, and it's just part of the virus life cycle.
Why is COVID-19 named COVID-19?
It's called COVID-19 because the COV stands for coronavirus, and the ID stands for infectious disease, and the 19 is the year it emerged.
And then the Stewarts asks, what factors change spreadability in viruses? Because influenza is R none, where measles is R none or 20.
It's the virus biology. So measles is an RNA virus, and it gets picked up really fast. And the faster the virus can replicate in the host, the faster it can be transmitted. So viruses have to overcome a lot of barriers when they infect you. Sometimes they need to go through they hijack your own cells and your own organelles to produce virus particles. So if a virus takes longer to replicate and transmit, it's less contagious.
Okay, I think I've reached the end of my list of questions. So if anyone else has any questions, you can put them in the chat for us. Sorry, that's my dog yawning. Alan asks, how are you filtering your own new information about COVID-19?
I look at the CDC and the WHO on a daily basis to get the numbers and see what they've found. I also, as a scientist, I go to the NCBI PubMed database, and that the PubMed website is for research that's been funded by the National Institutes of Health and other reputable governmental and federal organizations throughout the world. And so if the CDC or the WHO cites research, I go look at the source they use to see who produced it, how they produced that data, and most importantly, who paid them to produce that data. But I rely on CDC, WHO, and then the local, the state health departments, because you'll see with the COVID that transmission in Texas isn't anything like what they're experiencing up in New York. And so our health officials have access to data in numbers that we don't have.
Cool. Ruthie and Sophia asked, how can we slow the spread of COVID with tests?
Okay, so we've already, and we should all give ourselves a big hand, we've already slowed the transmission by going through this quarantine. So even though we're seeing infections in lots of numbers, they're not nearly as high as they would have been if we would have kept our normal lives. Testing is going to help stop the spread or help reduce the spread because we'll know when we have lots of these tests available, we'll know who's got acute infections, who's able to transmit virus, so we can tell them to stay at home. And we have that now with flu. You go to the doctor, you feel sick, they swab your nose, they say you have flu, and then you get your doctor's note, and then you don't go out. Or if you do, you wear a mask. So we'll have that available for COVID here pretty soon. Also, we'll have the antibody test to see who has had infection. And so if you've already been infected with it, you're probably not going to get infected with it again. And so you're going to know you don't have to worry about it if you start developing cold-like symptoms in the future.
Sarah asks, what determines how much the virus will replicate within a person?
Ooh, there's a lot of things. So viruses use you they hijack your ribosomes to copy their genomes. They hijack your endoplasmic reticulum and your Golgi to make their own membranes and proteins. And so depending on how large the virus is, how complex this genome is, sometimes they can even go into your nucleus and shut down your DNA replication. So, but the larger the virus is and the more complex it is, it takes it a lot more than a small virus, like say Zika virus or dengue virus. Within 3 days, you're already contagious and transmitting to other people. But within a week, that virus is gone. But then you have other viruses where your incubation period is like over a week to 10 days before you start getting symptoms, because it takes a long time for the virus to remodel your cell machinery to produce viruses.
Let's see. Someone asks, is everyone going to get COVID-19?
That's a tough one. Well, no, not everybody, but it is it's a pandemic, which means it's everywhere. And once a virus has reached pandemic level, you can't stop transmission. We can just slow it down. And so that's what we've been doing with this quarantine. We've slowed it down. And that allowed us to determine the resources we need. It's allowed us to accumulate personal protective equipment. It's allowed time for healthcare providers to develop protocols and researchers to develop diagnostic procedures. And so, yes, we are still, it's going to spread around more, but we're going to be able to address it a lot better. So the death rates, you're going to see them dropping. If you've looked at the data every day, the case reports and the death rates are reducing. And so it's going to circulate for a time. I don't know that everybody's going to get sick, but we are going to see transmission for quite a while. And we may even experience an uptick in transmission, but that's normal virus transmission. It happens with flu, it happens with other viruses. But just know that researchers and the doctors, we're all working to address it and make sure we have the resources available to take care of everybody who does get sick and needs hospitalization.
Cool. And will the coronavirus become like the flu where it reoccurs every year?
That's a hard one. It's this coronavirus is zoonotic, and so what needs to happen when it jumps species, it went into people, and now we need to know if it's going to be able to establish and become stable within the human population. And so when a virus jumps species and goes into humans, it can do two things. It can either not replicate in us for a while, but we're not the best host for it, so it will just go away, and that can take a couple months. Or it can be like, oh, I'm in humans and this is a really good host for me and I'm going to stay here. That's what happened with measles virus. So measles virus originally came from cows, from a virus called rinderpest. It jumped from cows to humans, where it evolved. It really liked being in humans and it turned into measles virus. Smallpox virus came from camels. if you can believe it, when we started living in cities thousands of years ago, and it liked humans so much, it mutated from camelpox into smallpox. So we do have, this happens a lot with flu virus where it jumps from birds. We actually share a lot of the same cell receptors for influenza as birds do, and that's why it happens more often. But for coronavirus, I don't know. It's different for every virus. And I hate having to give people that answer. But when we're talking about viruses, coronavirus versus flu, it's like we're comparing snakes with horses. They're both animals, but they're completely different.
Cool. Maddie asks, what is the shape or structure of COVID-19?
That's, it's an icosahedral symmetry. It's round. It has these spikes sticking off it that are bind to the receptors. So it looks spiky and like a crown if you look at it on the scope.
Katie asks, as public places begin to open up in Texas, how do you recommend people behave in those spaces?
First, you should know that directives have come down from the governments to tell these restaurants and these other facilities, giving them new policies and procedures for keeping things clean and reducing transmission of any sort of pathogen in their facility. So you should really know that they're doing their part to keep you safe. Also, wash your hands and don't touch your face. I can tell you that I cannot wait for restaurants to open up. I think my first place I'm going to go to is Moroso's. So I know everybody's doing their best. Yeah, but I'm still going to wash my hands. We're all going to be maintaining social distancing for quite some time, and that's it.
Cool. We have time for just a few more questions. So I'll ask Ellie's question. She asked, how long does the coronavirus last on surfaces?
How long or how does it?
How long?
Okay, so it depends on the surface, and it depends. So if you sneeze and your snot comes out and it's on a surface, actually coronavirus can live for quite a long time if you've sneezed it out with some mucus. If you've just like rubbed your eye or picked your nose and you touch a surface, or you're talking like I'm talking, it can live for a couple hours to a couple days. What really determines it is what is the surface and what's the environment like. If it's really dry out, if it's really hot or really cold, the virus won't survive.
Charlie asked, what's the difference between an epidemic and a pandemic?
An epidemic is when you have a virus outbreak within a small pocket of geographical location. So there was the Ebola epidemic that was only in a certain part of Africa. We've had Lassa epidemics, that's in a certain part of Africa. And we have measles epidemics when we have pockets of people who haven't been vaccinated. So that's in a small area. A pandemic is everywhere. Everybody's sick everywhere. The virus isn't confined to a small location.
There's a few questions about our pets. Are our pets safe? Can they get coronavirus?
We don't know. We have to develop the test to be able to test those animals. Coronavirus is a zoonotic pathogen. Cows have it, pigs have it, birds have it, bats have it. And if it can jump from a species, from like a bat to a human or a bird to a human, there's no reason why with its high mutation rate, it couldn't jump to another animal. Now, whether it causes disease or becomes stable in that animal, we don't know. We have just stories of people having cats or tigers or their dogs test positive for COVID. And that's just because viruses are everywhere and we get infected with all kinds of viruses every time we eat or breathe, and they may not cause disease. They may not even replicate to a high enough level to be transmitted. So yeah, I think I wandered. I'm sorry.
That's okay. A couple people asked more questions about the surfaces that the virus are on and the temperature. Tom,
Yeah.
can the virus be killed by freezing? And Ruthie asked, can the COVID survive in pool water?
It should be able to survive in pool water. That's got chlorine in it. And so that's gonna, so that's at a high enough concentration. If it can kill bacteria, it can pretty much kill a virus. When you have an enveloped virus like COVID, they're usually a lot easier to kill than the non-enveloped ones because it's really easy to denature or break up the protein on the outside. And if you disturb the outside proteins, like those spikes on the coronavirus, it can't infect. And so, yeah, pool water, freezing can kill some virus, but I routinely keep my viruses in the freezer. They're all enveloped. And I might see a small reduction in infectivity, but I still get virus out of it. So I would not use freezing as a means for disinfecting anything from a virus.
Okay. I'm going to ask two more questions. A few people have asked if we all go back to returning to public spaces, will there be a surge in the virus again?
We're going to see transmission, but we already have, hopefully by the time we go back to that, I'm sure you've read the reports that everybody's reopening in stages. And so what that's going to allow us to do is manage the transmission of virus as we start returning to normal. So if we get to stage 1 and we see a major uptick in transmission, it gives us the opportunity to go back to ground zero and rethink the way we're doing things. And so by the time we get to hopefully back to normal life, we're really not going to see a whole big uptick in transmission, but this is a new situation. We haven't dealt, the last pandemic of this magnitude was the flu pandemic in 1918. This is a completely different world than 1918. And so we're really learning as we're going. There are people trained in public health and disease transmission who are really doing their best to tell us how the safest way to go back to normal. But we're doing it in stages, so if there's a problem, we can always go back and try something else.
Great, and then the last question, what got you interested in virology?
Seeing a picture of a baby in Africa that had a tube going up its nose, it just looked terrible. Yeah. Seeing how sick they make kids, because fortunately, young people aren't very susceptible to getting sick from coronavirus. You can get infected, but the young people don't get as sick as old people. But normally, for most viruses, it's the very young children and the very old that have the worst disease. And it really bothers me to see kids sick and dying.
Awesome. Well, thank you so much for spending some time with us and with our guest. I learned a lot. I hope everyone else did too. So thank you so much.
Mm-hmm.
Bye, everyone.
Bye.